Hi, my name is Emily Neal, and I'm an instructor for Bio 161. And this week we're introducing microbial source tracking in Lab 10, where we talk about how to distinguish different strains of E. coli. This is important because water can be contaminated with fecal samples, and when that happens, it's unsafe. So you might have noticed these signs around on the beach. And these are posted when they find E. coli cells or other types of bacterial cells that are commonly found in feces. This indicates that there's some sort of fecal contamination in the water. So when that happens, when we find that, the signs are posted to prevent people from getting in the water, but the fecal sources could have gotten there from a number of different places. Perhaps from pets or dogs playing on the beach, from pigeons or other animals that happen to live near the water, like the pigeons pooping off the pier into the water. It could also be from local agriculture that when streams and runoff runs into the ocean, it takes that fecal samples with it. Or it could be from just human waste that also runs into the water. So we're trying to identify what the sources are of the fecal contamination in the ocean. But in order to do that, we also need to sample these possible sources. So students in the Micro 224 class are isolating E. coli from potential sources like dogs, pigeons, cows, or even humans. And in that class, they will isolate single E. coli cells and grow them up, making sure that they are single isolated colonies. Now, bacteria replicate through a process called binary fission, and this is really similar to mitosis in that you get an exact replica, an exact genetic replica of that cell. So at the end of binary fission, you end up with two genetically identical cells. After many rounds of replication, all of these cells end up growing together in a large clump, and this forms a colony. So at the end of Micro 224, they have isolated samples of these cells in these E. coli colonies. And just to keep in mind here, they are all growing on a plate. So one of these single isolated dots that you see on a plate is a colony, and a colony is basically a group of genetically identical cells. Basically, these are all clones of each other. So all the cells in a single colony are clones of each other. In Bio 161, what we're going to be doing is sequencing the E. coli cells that you'll be getting from Micro 224. Each group will receive a separate isolate. So for example, group 1 in a particular class might receive isolate number 97 from humans, which we designate as HU to stand for human 97.
This is a plate from the Micro 224 class, and now we're going to show you how this gets prepared for Bio 161. Here, a single colony is picked using a device called a loop. And then the bacteria from that colony gets spread over a new plate. Of course, you can't see individual bacteria here. Those bacteria need to be allowed to grow for 24 hours before you can see individual colonies. The plate is placed in a 37 degrees Celsius incubator to allow the bacteria to grow. 24 hours later, the plate's removed and the colonies are visible and ready to be used by Bio 161 students.
So in this example, right, you're going to get a colony from, or an isolate from Micro 224, this human 97, and each person in a group will be in charge of a particular clone or colony from their isolate. So human 97 might have 3 colonies, A, B, and C, and each person in a group will be in charge of one of those colonies. The way this happens is that the initial isolate that comes from Micro 224 is separated into single cells, and each one of these cells is allowed to grow into its own colony. But because it came from the original human 97 colony, all of those cells should still be genetically identical. So human 97A, B, and C should all be exactly the same. At this point, each student in the group will have sampled a different colony but from the same isolate. The next step will be to amplify and sequence the DNA of those different colonies, but they should be the same because they were from the same isolate. The instructions for PCR amplification and pyrosequencing will be presented in class, and portions of it will be in your lab manual as well, so we're not going to discuss the details of the protocol here. But eventually, the data that you are obtaining in class will be given to students in a bioinformatics class. They will be evaluating your data and uploading it to a research database. Basically, your data will be used for microbial source tracking, hoping that we can eventually make a match between the environmental sample and a source.